everyone. Thank you all for joining us for our June Research Spotlight webinar. I'm Dr. Sarah Hernandez, the Director of Research Programs at the Hereditary Disease Foundation. To start, I want to thank our sponsors for this webinar, Ionis Pharmaceuticals, Novartis, Argenta, and Bristol Myers Squibb. Also, a big thank you to everyone who supports the HDF. It is because of you that we are able to fund the exciting science that is transforming what we know about Huntington's disease. And of course, a big thanks to all of you for being here. Today's speaker is Dr. Walter Koroshetz, the director of the National Institute of Neurological Disorders and Stroke, known as NINDS for short. He joined NINDS in 2007 as deputy director and has been part of many NIH initiatives, including as co-lead for the Brain Initiative, the Blueprint for Neuroscience, and the Undiagnosed Disease Network. He also co-leads a number of the NIH Common Funds programs, including the Somatic Gene Editing Program. Before NINDS, Dr. Koroshetz served as the Neurology Vice Chair and Director of Stroke and Neurointensive Care and led neurology residency training at Mass General Hospital as a Harvard professor. His early research in the lab and clinic focused on Huntington's disease, where he was part of the team that performed the first study of pre-symptomatic testing. Today, we will be hearing insights from Dr. Koroshetz about 40 years of Huntington's disease research. Before I pass it over to Walter, I want to remind you all that you can ask questions in the Q&A box throughout the talk. So we should have about 25 minutes at the end for a Q&A. So send your questions in as you think of them. We don't want you to forget, and we will be able to address as many of those as we can. Also a reminder, this will be recorded and available on both our website and our YouTube channel in about 2 weeks. So if you want to watch it again or if you want to share it with someone, it will be available for that. Thank you all again for being here, and I will now pass it over to Walter.
So yes, I'm really pleased to be able to talk to you today. As Sarah mentioned, I'm the director of the Neurological Institute, and we're responsible for developing better treatments for people with Huntington's disease. And it's very close to my heart because, as Sarah mentioned, I was heavily involved in Huntington's research and the care of folks with Huntington's disease prior to coming to NIH. And we'll kind of revisit that story in a bit. And in terms of the research funding, my first grant was from the Hereditary Disease Foundation and the HDSA combined. And so I got my start really because of the funding from the HDF. So really happy to talk to you today, try and give back something for all you've given me. I'm going to show a couple of slides. So the NINDS I'll talk about first, and so we're the institute that has in our portfolio all these different neurological disorders. We kind of split some of the disorders of aging, like Alzheimer's and other dementias, with the National Institute of Aging. But we're the only institute at NIH that's 100% devoted to neuroscience. And we have some really serious problems that we're trying to work with, multiple different disorders that when you add them up account for significant amounts of mortality and morbidity in our country and around the world. The one good thing about NINDS that you may not be aware of is that by working in all these different diseases, we have this view of the world and science where we can kind of see relationships between different disorders. And so, for instance, our work in multiple sclerosis, which is the inflammation in the brain, has been very relevant to neurodegenerative diseases in terms of understanding the role of inflammation and other disorders like stroke or Alzheimer's, Huntington's disease even. We do a lot of work with rare disorders, which are mostly genetic disorders, neurogenetic disorders. And of course, as you can imagine, Huntington's being a genetic disorder, there's a lot of commonalities in how we approach those different diseases, particularly as it comes to genomic therapy, which we'll talk about. So at the NINDS, we have in this very unique position to be able to see science going on in all these different spaces that are really very interrelated. Also at NINDS, we're one of 27 institutes and centers, so we get to see and hear about research going on in places that you would never imagine. As a neurologist, it's really hard to keep up with just the neurology, let alone the neuroscience, which is real broad, and then all the other areas of science. And I think one message I give to people is that you can absolutely guarantee there are going to be major scientific breakthroughs that are coming that are going to be relevant for Huntington's disease, but you cannot predict where they're going to come from. They come out of the blue, and we'll talk about one, CRISPR gene editing, which came from work really trying to understand how bacteria protect themselves against viruses. So who would have guessed that the machinery that bacteria employing are now being used in people to rectify mutations. So that I think one take home point for people to bring away, that these discoveries in our ecosystem here in the U.S., the science is so advanced that breakthroughs are coming, at a really significant rate. And they're going to be relevant. You can't always tell in the beginning what's relevant, but but it happens. Okay. So our mission here at NINDS is to support research across from basic to translational to clinical research. We fund and conduct research training and career development programs to ensure we have a vibrant, talented, and diverse workforce. And we try to talk to folks about what we're doing so that the value of what we're doing is recognizable to the taxpayer, to the physicians, the patients. And so we're just today gearing up for a nonprofit forum we have every year. We have probably 150 or 200 different nonprofits who are disease related, and they all come in together and work together in one nonprofit forum. That's coming up next week, and I'll try and put a link in the chat if people want to register. It's available online. And there I was just working with the people going to be speakers, and heavily going to be towards, in the beginning anyway, towards how do we develop new treatments for neurological diseases, and people from all different industry, academia, outside of neuroscience, inside of neuroscience. So it's always a great program. The most important thing we do, though, is to fund research, and our budget for funding research, our kind of base budget, it has been around $2.2 billion a year. And some of that money goes to about 20 to 22% of it goes to really understanding how the brain works, how the nervous system works. That's really agnostic to any disease. Most of the money, about 50% of it would go to understanding mechanisms of disease. So how does the Huntington gene cause disease? That would be basic mechanisms of disease research. That's where most of our money goes. And in both those areas, it's critical that we fund this kind of research because it's the foundation of all the knowledge that then the industry can take to develop therapies for people. So that's where most of our money goes. But we do have a program that is called a translational program, and that tries to help investigators who are studying, Huntington's disease, for instance, and have an idea about a therapy. Now they're in, a university, they're not in a company, but what we've been able to do is to put together a program where they would have access to different companies that do pieces of the work that's needed to get a treatment to being tested, approved by the FDA to go into testing. So we're not a drug company, but what we try to do is develop therapies along the line where they're too risky for a company to do right now. But if we can show it looks good, that all the stars are aligned, that a company will then pick it up. And so that we call that de-risking the development of new therapies for different diseases. And and we have quite a bit of activity in the Huntington's disease space in those programs. And then we have a clinical trial group, and they do fund clinical trials around the country and those clinical trials are usually testing a therapy that could go into practice. So a lot of them are what we call comparative effectiveness treatments. There's a couple of things you could try, which one's best. So we'll set up a clinical trial to test one thing against another. Sometimes we'll actually test a new therapy, but that's less likely. That's usually up to the companies to do a new therapy that's then going to get FDA approved. We do more testing things that are actually FDA approved for other indications. Uh, and we have some of this is wide open, so an investigator with a good idea can come in and propose a clinical trial in patients. But we also have networks, and the one that's relevant to Huntington's disease is called NeuroNext. It's a network around the country, and its job is to test a new therapy that's never been seen in patients before uh, in any neurological disease, except for stroke, because we have another stroke network. But this network, say for instance, if there was a new treatment for Huntington's disease, and they wanted to test it to see if it had, early stage trial to know if it has a good effect. This network would be ready to do that, and it's run out. The center is out of the Mass General Hospital, but they have probably 25 sites around the country, and they'll do diseases that affect children, adults, very, very kind of broad abilities. Okay. Um, Wrong way. Okay. In terms of neuroscience research at NIH, one point to make is that neurosciences get more money than any other area, including cancer, including infectious disease, including heart disease. So most of the money at NIH, $11.8 billion, goes into neuroscience. The neuroscience money, though, goes to multiple different institutes. So at NIH, there's one cancer institute. They have most of the cancer funding. The neuroscience funding is split around many different institutes. The big ones are the Aging Institute with Alzheimer's dementia research money, NINDS. And IMH is the Mental Health Institute. The Drug Abuse Institute is NIDA, and the Eye Institute is NEI. They also do vision research inside the brain, so they do a lot of neuroscience too. Um, okay. And this is what I was talking about before about funding across the spectrum of basic science to translational science to testing new treatments. The goal here is not to have a gap. So you don't want to fund a lot of great research and then basically it's got nowhere to go, can't really help patients because it doesn't have a mechanism to go from the discovery on this bench to actually a therapy. Um, and so we try to keep keep that flow of information towards therapy as the major goal. We work with a lot of patient groups, as I mentioned, the nonprofit forum. We often meet with disease organizations, learn from them. We often do workshops or planning for research recommendations that involve patient groups, people who have the disease, or advocate organizations that represent people with disease, big parts to play there. Um, we have a program on Friday where we actually ask someone who has one of the neurological disorders to talk to our staff here. So many of them are PhD scientists and they have not taken care of patients. And it's really valuable to have patients tell their stories. They're incredibly motivating, and it gives our people a sense of the value of the work that they do here, which is mostly to fund the research that goes out. And the way that happens is that anyone in the US from a reputable institution, and that list is long, can submit an application for a grant. Then there's a group that gets together around that grant. It's called a peer review group, scientists who have expertise in the general area, and they review all the grants. Then they give the grants a score from 1 to 100, and then those scores are given percentiles, where the best scores are the first percentiles, then the second percentile, and then what we do at NINDS with the money that we have, we fund the best scores until we run out of money. And so right now we run out of money about the 8th percentile. So it's really not a good situation. It's hyper competitive. It means 92% of the grants do not get funded. So you'll hear from investigators that NIH doesn't like us, they won't fund our grants. It's not true. It's just that they just didn't make the pay line, which is now unfortunately at 8th percentile. And that's just arithmetic. It's just the amount of money that Congress gives us divided by the number of grants times the cost per grant. The average cost per grant now is about $550,000 a year for the regular grant. So one grant is over $2.5 million. And so that's really how we work. It's a very democratic American system. You know, it's not top down. I don't make decisions on what gets funded or not. It's the peer review, the best scores. We fund them until we run out of money. Um, so it's a very democratic. It's not a perfect system. The peer review system, you know, the people are doing their best, but they're not perfect. Um, but certainly, if I go around the world and see what other countries are doing, it's really the best system. It's not really who you know. It's what your how your idea stands up to the competition. Um, okay. Um, Okay, now I was going to just go back and talk a little bit about, you know, how I got to where I am because it might be of interest. As I mentioned, I'm a neurologist and I trained in neurology at the Mass General Hospital. And when I finished my training, I was basically going to go work in the laboratory to understand um, how neurons function in terms of their very basic properties that we call channels that are in the membranes, and the channels open and the cells fire. There were new technologies that just come out, and really excited about that. My chairman is seen here, Dr. Joseph Martin. He's sitting on the stairs here, and basically, you know, what we usually did in those days is we went into the laboratory, and then half a day a week we would go to clinic and we would specialize in a particular area. So most of the time we're in the laboratory, but we would, you know, you could become a really good clinician if you just focused on one area. It would be hard to be a very general beat the general neurologist over time if you were spending most of your time in the lab. Anyway, I was in the elevator with Dr. Martin one day, and he asked me, "Well, what are you going to be doing?" I told him about the lab. He said, "Oh, that's great." And he said, "What are you going to do for your, you know, one day clinic?" And I said, "Well, I haven't really decided yet. I've been thinking of a couple of things." And he said, "Well, why don't you just do Huntington's with me?" So I said, "Okay." So that's how I got into Huntington's disease. So serendipity is not to be ignored, and also the force of people. You know, if Dr. Martin asked you to do something, you knew it was going to be something good, and so you tend to say yes. And that's true about lots of my career. I ran into people, they were good people, they asked me to do something, and it sounded good, so I did it. So, uh, and that was kind of the beginning in Huntington's disease. And then, so in the Huntington's clinic, it turned out that Dr. Martin had just hired a whole bunch of people to focus on Huntington's disease. He got Ted Bird here, you can see here working with the dry ice. Ted was an endocrinologist. Dr. Martin was actually a neuroendocrinologist. Ted Bird was interested in Huntington's disease and the endocrine abnormalities in Huntington's. And he was also very interested in understanding what was going on in the brain in Huntington's disease. So he built a brain bank and he started a clinic, and I joined that clinic. And in the clinic was a guy named Richard Myers, who's seen here, and he was a geneticist, a clinical geneticist, and also at that time, Dr. Martin hired Jim Gazella, who's seen here. And Jim was a graduate student at MIT working with David Hausman on genetics. And they said, well, we think if we'll put Jim on the project of identifying the Huntington's gene. So he came to Mass General and he recruited Marcy McDonald, who's seen next to him, and they went after trying to find the Huntington's gene. And then Rick and I were basically in the clinic collecting blood samples from patients and going around the country trying to find insightful cases to try to help find the gene. And then we were really surprised that we were able to narrow down the chromosome and the genetic area where the Huntington's disease was, and I think that was in 1990, probably 93. But it allowed us to do pre-symptomatic testing for Huntington's using that genetic information. We would have to get the genes from your parents, your siblings, the unaffected, the affected siblings, and then we would be able to, with pretty good confidence, let people know if they inherited the Huntington gene. So as you can imagine, that was early stages. It's a lot of really intense discussions with people who are at risk for Huntington's on whether it was the right thing to do to have the testing or not. But I think we did a good job in trying to make sure people, when they actually did get the result, that they really wanted to know the answer, whether it was a positive or a negative result. So very interesting times there in the lab, and then I also was working in the laboratory with Flint Beal, who Dr. Martin had brought in to work on Huntington's disease in animal models. And what we had found is that there are certain toxins you can give to the animals and put in their brain, and it would look a little bit like Huntington's disease. That was the best we could do in those days. We didn't have transgenic animals. But what we found was that there seemed to be a mismatch between the energy that was available and the energy that was needed. And in those cases, the cells would produce lactate in the brain. And so we decided, well, let's see if that's true in people. And I worked with this fellow in the lower corner, Bruce Jenkins, who was a specialist in MRI spectroscopy. So MRI scanning, you think of pictures of the brain, but spectroscopy, you basically get measures of the chemicals in the brain. And so we looked and we saw that people with Huntington's disease had very high lactate levels in the brain. And then we went about trying to lower those lactate levels, thinking that would be helpful to patients. And so I spent a number of years doing that, and actually that turned out not to work, that we were not able to find the drugs that could lower lactate. We could find drugs that lowered lactate, but it didn't seem in the clinical trials. And we did two really big ones, both funded by NIH, and they just did not show benefit. So story there is a lot of work with trying, and it doesn't always work in the end, and that's what research is all about, and you just can't give up. You got to keep trying something new. And in trying that, working through that, I think that I just wanted to mention Nancy Wexler. I'm sure everybody knows Nancy here, but she was a real pioneer in getting Huntington centers around the country. She's seen here in the middle testifying. That's actually Francis Collins with the glasses, and she was just an amazing motivator in terms of the Huntington space. She was actually started at NIH. She was a program director here in our institute years before I got here. And then she went out and started the foundation and worked really hard to track down the gene. And really her work in Venezuela is what led to the big discovery of the Huntington's gene. And So really great. Now she unfortunately is suffering with Huntington's disease, but it's just the power of one person. The power that one person can have is just astronomical. And Nancy is a great example of that. So these are the kind of the papers that I was involved in, the predictive testing. We have a paper about the genetics of doing the testing, and then all the work we did on trying to lower the lactate to improve outcome for patients. In the first study, it looked like there was a difference. There were It wasn't a big difference. The second trial, though, did not see, we couldn't confirm that difference. So we lost out on that one. So a lot of things that, I did when I was at Mass General Hospital, I was in the Huntington's clinic. And I also, interestingly, would go around with Rick and Carol Mastromoro was the social worker to different, long-term care facilities where Huntington's patients might be, in the last stages of their illness, being cared for in one of these skilled nursing facilities. Um, and one of the days we went up to what's called Lowell Healthcare in Lowell, Massachusetts. It's about 25 miles north of Boston, and they had, I think, a patient with Huntington's there, and we kind of talked to them about care for people with Huntington's. And the person who ran the healthcare facility became really interested in this, and he took it as a vocation almost. And eventually, I think the top number, I think we had 44 people with Huntington's disease brought being taken care of in this healthcare facility. So I was the doc, and I would go up at least once a month for 17 years until I came to NIH, taking care of people with Huntington's disease and kind of the later stages of their illness. But another example of serendipity, just being able to tell a story and people buy into it, and put together a really good program. So Jim was really good at doing that. They did some really innovative things in the early days to help people with Huntington's. Okay, but as I said, things didn't work out as I wished. I wish I had a treatment for Huntington's, and it looks like it wasn't going to happen in that short time frame I had. So I thought that going to the National Institute of Health would give me the opportunity to have kind of a broader impact on the people that I couldn't help while I was at Mass General by working nationally to push research in neurological disorders. So that's when I came to NIH, and this is the hospital at NIH. So there's most of our money, 90% goes out to the universities, but about 10% stays with the research that's done on the campus. And there's a research hospital here. Have not been that active in the hospital, been primarily active in kind of the overall running of the institute. In terms of Huntington's disease, so we found the gene. We realized early on that the CAG repeat was the length of it was related to the time of onset, but then we kept learning new things, and particularly now the work that Jim and Marcy continued to try and find out what genes do we inherit that affect when you get Huntington's disease or how fast the disease progresses. And they found some really interesting genes that modify Huntington's disease. So besides the CAG repeat, there are other genes that affect how Huntington's starts and progresses. And it turned out that a lot of those genes had to do with DNA repair. And not entirely obvious why that would be, but we'll go on and we'll find out that some interesting things came out of that. Let's see. And that has to do with the fact that the repeat which you inherit, the CAG repeat, normally you can get up to about 35 or so and not have any trouble. Between 35 and 40, sometimes you get old onset Huntington's disease, and then after 40, it's more regular. Some people with really high repeats can unfortunately get Huntington's when they're children, even as children. And that's what you inherit. So that's what the number that's floating around your bloodstream, and that's where we do the blood test to look at the gene. But what we found out, or not we, but what investigators found out from the research that we funded, is that the CAG repeat is changing inside the body even after you inherit it. And that was really remarkable. That's called a somatic mutation. So it's not a germ cell mutation. It's not in the sperm or the ovary. It's something that's happening in the cells. It also seems to be happening more in the areas that are heavily affected by Huntington's disease, i.e., the basal ganglia. That's where you see even the bigger somatic mutations seen here in this middle box compared to what you'd see in the tail of this is a mouse, or the liver of the mouse. And uh, and then these DNA repair enzymes that that Marcy and Jim and others discovered, but it was found out that they modify how the mutation expands in these tissues, as you age, and and that is really interesting because the next thing, going the wrong way. The next thing that just came out is that there's some evidence that the tissue can handle the CAG repeats that you inherit. So if you inherit like a CAG repeat 40 or 50, you're going to get Huntington's disease. The cells are going to die, but the cells that die don't have 40. They have the big expansion. They might have 120 repeats. So you inherited 40, but in the cells in the striatum, it'll jump over time to 120, and that's when the cells start to die. That's a theory that's just come out in the last, say, 6 months, and a lot of evidence for it. We'll have to wait and see how it holds, whether it's watertight. But the important thing is that these other modifier genes, they prevent that from happening. So if we can then go in and manipulate those modifier genes to prevent the expansion, there's a lot of hope that that's going to basically prevent Huntington's from actually happening, even though you inherit the gene. So that's really the excitement now going forward, that we potentially be able to do this. Now, how do you do that? Well, we have a couple of ways now, and the one I wanted to talk about is gene editing. So this is a discovery I mentioned right when I started. This came from studies out of MIT and UCSF, and how bacteria deal with viruses. Basically, when the virus comes, it gets its DNA information into the bacteria, and then the viruses start to expand. They kill the bacteria. Well, the bacteria have a way of searching for those, and they cut them out. They cut that information that came from the virus, they cut it out. And that's what CRISPR is. It's an editing tool that cuts out certain stretches of DNA. But now you can do it for all sorts of things. You can do it to change the expression of genes. You can cut out DNA and potentially cut out the Huntington gene, or you could cut out one of these genes that then these DNA repair enzymes that are responsible for the increase in this growth of the CAG repeat as you age. So this is new technology. I'm running with a colleague here a somatic gene editing program, and we're actually doing some of the first gene editing in people now. And there was recently a New England Journal of Medicine paper of 2 kids where the mutation was found and was treated within 7 months with gene editing. That was in the liver, but we have really good technologies now that are not right there, but they're really, really close to being able to do that in the brain. And that a lot of those technologies came out of what's called the Brain Initiative. But these new technologies, the genetic sides are very precise. They they do exactly what you want them to do, affecting the gene. They're much different from drugs, where the drugs go everywhere and they do a whole bunch of things. These therapies are just going to be like, really very precise. And and I think that's kind of probably where I should end on right now. I think we have a lot of other programs, as I wanted to mention here, that have lots of parallels with what's going on in Huntington's, particularly the Brain Initiative. There's a lot of those tools to get genomic therapy into the brain are coming from the Brain Initiative. But I just hope that was interesting. There's lots of really cool stuff that goes on here at NINDS, from understanding how brains work, how they're put together, to how to develop treatments for different diseases. So why don't I stop there, and then I'd be happy to talk to folks about whatever their interests are.
Excellent. Thanks so much, Walter. And I love that brain you showed at the end. I did some fruit fly work at the end of my postdoc, so I have a soft spot for flies in my heart. So I think I'm going to start at the top of the question queue and we'll go down. So people are essentially interested in clinical applications as well as trials. So someone asks, what do you think will be the first clinical trial which would be approved by the FDA and EMA?
Well, I can't really tell you that. I don't know the answer. I think probably the things that are, right there that might come out first. And but you have to, as I said, I spent probably 15 years on those trials which didn't work, so you can't tell until it works. But certainly the beginning of ASO, the antisense therapies, which were put into the spinal fluid, and they block the expression of the Huntington gene. Those were, I think, we all had a lot of hope that those were going to work. Now they ran into some issues, and it may be that those can be overcome, but those are probably the furthest ahead right now. Those The newest things are maybe not necessarily ASOs, but interference RNAs, which do somewhat similar things. May get away without some of the toxicities. The big problem that I think we're running into is that the Huntington gene, if you don't have either copy, then you don't live. You don't get born. It's lethal, embryonically lethal. So the normal Huntington gene is important. So what we really do need is a therapy that targets the Huntington mutation and leaves the normal Huntington gene alone. That would be important if we could get there. That might be the problem with the antisense therapies that have been tried so far, is they don't do that. But then there are also people working, to tweak these modifiers to prevent the somatic expansion using similar, interference RNAs. And then you have the gene editing folks. So I didn't mention this, but the lady and the woman scientist at UCSF who got the Nobel Prize for gene editing, we actually fund her to gene edit out the Huntington gene. So she's made it. I'm not sure. I would love to talk to her, find out how she got onto this, but that's one of the projects that she took on in her laboratory, and this is the Nobel Prize winner for gene editing. So you can't do better than that. And so she's working, and her lab's working on editing out the Huntington gene. Um, so I think I would say that I move more towards optimism on the genomic side of things than on the drug side of things.
Awesome, thanks. So this is someone who said their family recently got a diagnosis of Huntington's disease. So firstly, so sorry, that's incredibly tragic. But I think my second point would be, you're in the right place. Thank you for joining the HGF webinars. Hopefully you learn a lot. If you have other questions, my email's on the website, reach out to me. Their question is around alcohol consumption and the progression of Huntington's disease. Do you have a recommendation there?
Uh, well, we actually looked at um, years ago with Rick Myers. We looked at modifiers, things like alcohol and smoking. Nothing was really strong there. Um, but I would say that, one of the issues that folks with Huntington's run into is that a part of the brain which is involved in kind of maintaining kind of your emotional equilibrium, that those circuits weaken in Huntington's disease, and alcohol really weakens those circuits. So the combination is not good for sure on the behavioral side. In terms of whether it makes the actual disease process worse, I don't have any information on that, but certainly a lot of the behavioral issues will be definitely worsened by alcohol. Yeah.
There was actually a really interesting study that just came out. It looked at neurofilament light, which is a biomarker, I'm sure you know, a biomarker for brain cell death. And it
Uh-huh.
showed in healthy people. So without Huntington's disease, people who had better cardiovascular scores had reduced levels of NFL. And so I think there's a lot of research coming out right now to show that the healthier you are, the healthier your brain is. Things that are just good for you and your body are good for your brain. So,
Yeah, I like to throw out that I forgot to say one thing, which is I'm not sure how well this has been documented in the literature, but with Dr. Bird, what we found was that people with Huntington's disease are using incredible amounts of energy, and these are folks who have chorea, but maybe not just only chorea. So Ted was a neuroendocrinologist, so he was interested in why the metabolism was souped up. But what we did find, we would bring people into the hospital who had lost tremendous amount of weight because they weren't eating to keep up with their energy. And we would give them really high calories. I mean, talking 3,000, 4,000 calories a day, and they would gain weight and their career would get better. So what I would always tell patients is to really watch your weight and don't let your weight drop. Try and stay, if you can, a little bit on the overweight side if for folks who have Huntington's disease. The opposite of what I tell people who are at risk for stroke. Yeah.
that's interesting. So we have a few questions around brain imaging. So somebody is asking what it would take for the FDA to agree to use brain imaging data as either a primary or secondary endpoint in HD therapeutic drug trials. And kind of the point that they make is that there's such phenotypic differences across even families. It's difficult to assess these different changes. And so I think overall what they're asking is, the FDA isn't using this and it's hard we don't have trials for pre-symptomatic people, and those may be the only changes we see. So what are your thoughts around using MRI images or brain images for primary or secondary endpoints?
Right. Okay, well, good question. So we funded a study called Predict HD, which looked at pre-symptomatic folks and tried to understand what would kind of be the earliest stages, and that it was imaging along with that. So there's a lot of evidence there that that you could use imaging to see changes over time. Now, my sense is that the value of that is not necessarily on the FDA side. We'll get to that in a second. The real value of that information is for the pharmaceutical and the biotech companies to work with patients to try and see if their drug is actually slowing down those kind of changes. Now, the problem oftentimes is that you have a treatment, and to know if it works, you'd like to have as much evidence as you can before you go into a big multi-million-dollar trial, and that's what you get out of a phase 2 trial. So in a phase 2 trial, you have to use these kind of endpoints like how much change is there in the striatal volume or how big the ventricles are getting. So I think that's where the real value is. Now the FDA, they're approving drugs for everybody to take every day, and they don't really care too much about things like that. What they care about is, does it make the patient better? And that's probably a good thing, because I've seen certain cases where it made the laboratory test look great, but it didn't help the patients. So you really want them to focus on what's helping patients. And that being said, it's really important for patients to come together and work with the FDA and inform the FDA of what's really valuable for the patient. And that really changes disease to disease. And it's very surprising in my field that what's really important to patients is oftentimes not what you would think of intuitively. So, one example, spinal cord injury. People are paralyzed. Everybody says, well, of course, they're going to want to walk again, so the outcome should be walking. And the patients say, no, what I really want to get first is bladder control, bladder valve control. That's what's most important to me. Unless you talk to them, you would, but that's really what's important, and it is important. And so I think that's where you want to interact with the FDA on what is important for patients. These biomarkers are incredibly important. So if you have 10 therapies, which one is going to go into a multi-million-dollar trial? You need some way of knowing if the therapies are really doing something good, and that's where the imaging could be really important. I worked heavily in the imaging space. I think Diana Rosas at Mass General continued that over the years. But there's really good data now on changes over time that could be used in any kind of trial to see if there's something that looks like it's working, prevent the atrophy. Yeah.
Yeah, I think I would add to that too. Brain imaging can be a little bit tricky because you want to show you're not just like overall brain volume isn't there because of neuroinflammation. And that's something that you can't do alone. You would have to do it in conjunction with other biomarkers. So I think as a primary endpoint, it probably wouldn't get approved unless there was something else to indicate that the preservation of brain volume isn't just neuroinflammation.
Right. Right. You could get fooled easily.
So here's one about the 8% funding rate that you mentioned. They say with only 8% of grants getting NINDS funding, how many of those are related to HD?
I don't know the number, but I did check before I got on that we spend about $44 million a year on Huntington's research. So if the average grant is 550,000, probably again about 80 grants, be my guess. And
Thanks.
it's not enough, that's for sure. It's true about all
Yeah.
of the diseases. Yeah, it's not enough.
And I would plug if anyone's interested in seeing the types of HD grants that the NIH funds, they have a fantastic website called NIH Reporter, and you can play around with their data, and it's really interesting. You can do it by region and by disease and all sorts of things. So really good website, NIH Reporter.
Yeah, everything's
Here's
public.
one. Yeah, everything is public. So here's one kind of related to that. They say there's been a lot of new findings in clinical trials in HD in the past few years. Despite HD being a rare disease, what is learned is likely to help other diseases. Is there any priority for funding around that?
Uh, well, as I said before, the way I run NINDS is to try to do as top down as possible. So I try hard not to say what NINDS priorities are. Instead, I feel that I have to rely on the study sections, who are the experts, review the grants. But we also have groups that will develop, research recommendations. These will be outside groups. They decide what they think is most important. They're, the experts, and they'll have people with diseases on the board. And those are for the whole community. So they don't dictate what we fund, but they're informative to people in the field to know what the experts think is most important. So we do more kind of soft prioritization as opposed to say hard prioritization, which is the opposite of what they do in China. They say you're going to do this and don't do anything else except this, and we're kind of the opposite.
Thanks. So here's someone interested in different models. What do you think is the best research model to pursue Huntington pathophysiology?
The best model is the human, but some things you can't do in the human, and so I think where we have with the genetics a plethora of models to work in, and really the model you pick depends on what the question you're trying to answer. So Sarah mentioned you worked in Drosophila in the past. So Drosophila is great for screening things because you can create the mutation, you can have thousands of flies, and you can expose them to 1,000 things, and you get an answer in, I don't know, 3 months, Sarah,
Oh,
something like
faster,
that.
2 weeks, 2
Yeah,
weeks. They're super fast.
2 weeks. So for some things, like a screening, you want a model like that. Or some people now are using iPS cells. So these are cells that are taken from a piece of your skin, and then they can be treated with a whole bunch of different genomic um, therapies, and they can be turned into neurons. So you could have a Huntington's neuron or a Parkinson's neuron, and then you can grow them up into uh, culture dishes. We have here a program where we have robots that are basically uh, feeding and examining thousands and thousands of these wells of iPS cells, and you can screen things there. And then a lot of work is done in the mouse models, um, because there again you can introduce the genes, you can do genetic manipulations pretty quickly, not certainly slower than Drosophila. And then after that, it's really a large brain model, and there you need large brain models. For instance, If you're interested in a therapy that you're going to give to people, it's got to go across the blood-brain barrier. The mouse brain is pretty small. There are sheep models of Huntington's. I think in Australia they had sheep models. Non-human primates are often as absolutely essential because they're closest to the humans. I remember in one of our projects, it was a pain project. The drug looked great, fantastic, and the mouse was fantastic. Non-human primate just killed the liver, and so that killed the program. At least it didn't kill the people, but no one would have known unless you did the non-human primate that it was liver toxic, because some things that just only occur in certain species. So, and then it's always a risk. Anybody who goes into a trial in Huntington's disease, they'll be talking to you about the risk because they're going to be trying things that no one's ever done before, and they don't know what's going to happen oftentimes. They try and mitigate the risk. They watch you carefully. Look like there's something going awry, they'll stop the trial. But it's but people with Huntington's disease, that without them, there's no treatment, and they're heroes in volunteering for these things because there is risk there.
I think that might be a good segue for one final question. And someone asks, how would someone get involved in clinical trials?
Great. Well, I think that there's a website. I wouldn't advise going to it right away, but there is a website called clinicaltrials.gov, and it has every single trial that's done in the US and many that are done in Europe and other countries. All the US trials have to be listed on this website, clinicaltrials.gov. You put in Huntington's disease, you'll see a whole list of trials. Now, the problem there is it's not clear what's best for you. So there, I think the way to do it, and Sarah, I know you guys have ways in which get information like that, or HDSA does, but also the docs who take care of people with Huntington's, particularly the docs who specialize in Huntington's, they usually know every trial that's going on around the whole world, and they know which ones are which, for which type, whether you're pre-symptomatic, symptomatic, early stage, late stage. So I think that kind of way is probably the best, is to, talk to the docs that are Huntington's experts. But I don't know, Sarah, what do you say to that one?
Yeah, I also always tell people to look at Enroll HD. So that's the largest observational trial of any Huntington's disease out there. And I think it has over 23,000 people currently enrolled, 75 sites worldwide. And oftentimes what clinical trial, what will happen is when people are looking for participants for clinical trials, they'll see who's participating in Enroll. Their doctors will also know more about HD clinical trials. So I would say that Enroll HD, your neurologist, and clinicaltrials.gov. Those are the best resources out there.
Great, that's true. Is that the CHDI?
It is, yeah, yeah, funded through CHDI.
Okay, yeah, that's good. That's good. Good advice.
All right, so we are coming toward the end of our time, so I'm going to wrap it up there. If you had a question that we didn't get to, I'm going to send those over to Walter. So if you left your information, he can follow up with you. I want to end by again thanking our sponsors for this webinar, Ionis Pharmaceuticals, Novartis, Argenta, and Bristol Myers Squibb, as well as everyone who donates to the HDF to support the research that we fund. And of course, everyone for tuning in. Last but certainly not least, our speaker, Walter, thank you so much for taking the time to tell us about your amazing career in HD research.
It's been a great career. I just wish we got to get a treatment before it's over for me anyway.
We will. We'll have you back when we get one.
Okay. All right. You guys take care.
Our webinar series is going to take a brief pause for the summer, but we will be back on Wednesday, September 17th. We're going to be joined by our 2024 Transformative Research Award winners, Doctors Andrew Yu and Osama Aldalama, who will be sharing their work for which they won the award, using stem cells and genetic messages to transform HD research. Registration for that webinar is now open, so right after this, head over to the HDF website so you can get it on your calendars. We are so proud to showcase the advancements in HD research through our Research Spotlight webinar series. So if you find these sessions valuable and you want to support our mission, please consider donating today. Thank you all for being a part of our community, and I hope to see you next time.